Okay, so in today's class, we are going to study that how carbohydrate digested in humans get digested in humans. We are going to study the detailed mechanism or detailed process of this carbohydrate digestion. Fine. So what
Yes,
happened?
ma'am.
First, the food enters into the mouth. Food enters into the mouth. Okay. So as I told you, salivary gland present near the mouth, and salivary gland is exocrine gland or endocrine gland. Tell me.
Salivary gland.
I told you it in my previous class. Hello.
Yes, ma'am.
It is exocrine gland. It passes its secretions to the mouth with the help of a duct. Fine. So as food enters in the mouth, salivary gland present in near the mouth secrete saliva in mouth. Secrete saliva in mouth.
Yes, ma'am.
And even the saliva also gets secreted because of the smell of the food. Yes or no? When something
Okay.
tasty is
Yes,
near
ma'am.
us, tasty food near us, so saliva automatically comes in our mouth. So the salivary gland get activated by the presence of food in mouth, by the smell of food in mouth, or many things, many such factors. Fine. So what is the function
Okay,
of
ma'am.
this saliva? First of all, the teeth that are present in the mouth chew the food. Chew the food. And tongue that is also present in the mouth, it helps in turning of food. Turning of food. And as the food turn, the saliva get mixed. Saliva get mixed with food. Fine, saliva gets secreted from the salivary gland. Now teeth are chewing the food and tongue is helping in movement of the food. So when the food moves, saliva get mixed. Is first point get
Okay,
clear to
ma'am.
you?
Yes, ma'am.
Okay. So in the second point, what happened basically? Now we have what? We have food which is chewed food. Chewed food means small particles of food. Fine? Plus saliva. So we have these two constituents in our mouth now. Okay. As I told you, saliva contain an enzyme that is called as the salivary amylase. Amylase. Now amylase do the reaction with these small particles, and what constituent of these small particles? Carbohydrate. Starch. This is a carbohydrate. Amylase get reacted with this starch that is present in the food particle. So now what is going to do? It is going to hydrolyze the starch. So saliva hydrolyze starch and convert it into maltose. Maltose.
Okay, ma'am. Yes, ma'am.
Okay. First thing that I told you, saliva contains salivary amylase. Saliva also contain some mineral salts. Mineral salts that help in maintaining the pH. That help in maintaining the pH.
Yes.
pH is very important. Why it is important? Because enzymes get degraded at the lower pH or they can get also degraded at the higher pH. Enzymes have a specific pH
Yes,
value
ma'am.
upon which they work. Like hydrochloric acid. Hydrochloric acid is very acidic. So it works only at 2.5 or 3 pH. It is not going to work at high pH value. Okay, so this pH is also useful. The salivary gland
Okay, ma'am.
secretes salivary amylase, which has some specific value of the pH. So pH also be maintained. pH should be maintained, and this pH is maintained by the mineral salts that are present in the saliva.
Okay, ma'am.
Fine. So the pH value upon the salivary amylase work is around neutral. It is around 7. It can be 6.8 or it can be 7.2. So it is around 7, or exact it can be 7. So it is around neutral. Okay. So have you understood the second process? Now we have saliva and food particles in our mouth.
Yes.
Now saliva has salivary amylase which act on upon the starch and convert it into maltose. Saliva also contains some mineral salts that help in maintaining the pH. And the pH value upon this, the salivary amylase work is around 7.
Okay, ma'am.
Or more basic, then it will not work. The enzyme get degraded. Fine. Now, next thing that is going to happen now is, now food, the role of mouth is now over. Fine. So food passed to stomach. To stomach. Through what? Through esophagus. Esophagus is called as the food pipe. So through food pipe, food get passed to the stomach. Okay. And in the stomach, we have HCl, hydrochloric acid. And this hydrochloric acid is very strong
Yes,
acid.
ma'am.
So we can say in the stomach or inside the stomach, the pH is acidic, or there is acidic environment.
Yes, ma'am.
Now see, we have
Yes,
swallowed the food. Our food contains saliva. Yes or no? When we swallowed it.
yes, ma'am.
Okay. But saliva work at neutral pH. It do not work at acidic pH. So what will happen when saliva comes into the stomach? Because of this low pH, salivary amylase get degraded.
Yeah, ma'am.
Because salivary amylase only active at the neutral pH. But in the stomach now, pH is acidic. That's why the salivary amylase get degraded. That's why pH is very important. Okay?
Yes, ma'am.
Okay. So salivary amylase get degraded because of this acidic pH, and this acidic pH is governed by this HCl. So this HCl, this acid denature the amylase. Denature means the amylase is lost its nature, or it get degraded.
Break. Yeah.
Okay?
Yes, ma'am.
Okay. If now amylase get degraded or get denatured, so what will happen? Starch hydrolysis stop. Stop now. So are you getting it? In the mouth, when pH is around 7, salivary amylase is working. Some of the starch get digested and converted into the maltose. But whole of the starch does not get digested because we can't put our food for long time in our mouth. Yes or no? We do not generally put food for long time in our mouth. So all the starch didn't get digested. But when food passed to the stomach, this amylase has been degraded. But all of our not digested. So there is another amylase that comes into small intestine. I will tell you the later. Okay. First, try to understand this, that in the stomach the salivary amylase get degraded, so starch hydrolysis stop. Suppose in the food particle 50% starch has been hydrolyzed in the mouth. Now 50% starch remains. It didn't get digested in the mouth. So it will digest further. But by not this amylase. The salivary amylase now get degraded.
Okay, ma'am.
Okay. Okay. So this here the role of the stomach has been completed. So what will happen now? Food get passed to the small intestine now. Passed to small intestine. Yes or no?
Yes,
And I
ma'am.
told you pancreas, which is a gland, secrete pancreatic juice, and it by a duct these juices, which are basically enzymes, to small intestine.
Okay, ma'am.
Fine. So in the small intestine, our food are partially digested food. It is partially digested. It is not completely digested, but partially digested food that comes from the stomach get mixed with this pancreatic juice. Yes or no?
Yes, ma'am.
Okay. So in the next point, what will happen? Now this pancreatic juice also have amylase. It also have amylase. And this amylase is called as pancreatic amylase. So we have two kind of the amylase. First one is the salivary amylase. Second one is the pancreatic amylase. Salivary amylase get secreted by the salivary gland, which act in the mouth. And this pancreatic amylase secreted by the pancreas, which act in the small intestine. Fine. So the starch that remained here, I told you the 50% starch has been remained from digestion. So the remained starch get hydrolyzed here. Because of this pancreatic amylase, and it get converted to the maltose. So now our 100% carbohydrate get digested.
Okay, ma'am.
Okay. Now see,
Mm.
the pH of the secretions of the pancreatic juice, pH of these secretions is basic or alkaline. Alkaline means basic. Okay. See,
Yes, ma'am.
from the stomach, food come into small intestine. But in the stomach, food get mixed with the HCl, which is a strong acid. Yes
Mm.
or no? So now food is also acidic.
Yes, ma'am.
When this acidic food come in the small intestine, so these alkaline or basic pancreatic juice make this acidic food neutral. Do you know base plus acid make a neutral?
Yes, ma'am.
So this pancreatic juice is base and the food contain HCl, which is acidic. So they both interact with each other and they make the food now neutral.
Okay, ma'am.
Okay, and at this neutral pH, amylase function. So this amylase that is secreted by the pancreas, it function at this neutral pH. That's why these pancreatic juices are basic, because they have to make the neutral conditions so that amylase can work upon.
Okay, ma'am. Okay, ma'am.
Can you relate this now?
Yes, ma'am.
Okay. So the role of the small intestine has been okay. Just one also thing remain. Now carbohydrate get converted into the maltose. It does not convert into the glucose. We want it converted into the glucose. So we want further degradation of this maltose. Yes or no?
Yes,
So
yes,
the
ma'am.
lining of intestinal wall secrete one enzyme which is called as maltase. Maltase. And which portion of the intestinal wall? Ileum. Ileum. So lining of the ileum
Yes.
is the part of the small intestine. So lining of the ileum secrete maltase. I told you na in previous class that if it is the duodenum, which is the upper part of the small intestine, and this is the ileum, which is the lower part of the small intestine. So pancreatic juice comes into the duodenum. Here starch has been converted to the maltose. Now maltose comes to the ileum. There is no starch in the ileum now. So this ileum now secretes this maltase enzyme in order to convert this maltose.
Okay, ma'am.
So lining of this ileum intestinal wall secrete an enzyme which is called as the maltase.
Okay, ma'am.
Okay, but now there is twist in this story. Suppose this is the ileum. Okay, this wall of ileum has some kind of the glands, which secrete this maltase enzyme. But this maltase enzyme did not come from this gland into the lumen. It did not come into the lumen. And it remain attached to cell membrane of this gland. It remain attached to this gland. So maltase enzyme do not secreted into the lumen. It remain attached to the surface or lining of that epithelium. That's why
Okay.
this enzyme is called as the membrane-bound disaccharidase, because it is the enzyme. That's why we call it as it disaccharidase. Okay, and why we call it as disaccharidase? Because it is converting maltose
Glucose.
into glucose, and maltose is what? Disaccharide.
Glucose.
That's why we called it as disaccharidase. So
Okay,
maltase
ma'am.
is a membrane-bound disaccharidase. Why it is membrane-bound? It is not going secreted in the lumen. It remain membrane-bound. That's why we call it as membrane-bound disaccharidase. Fine. So what this
Okay, ma'am.
maltase is
Yeah.
going to do? So this maltase convert maltose into glucose. And what kind of the glucose? Alpha glucose.
Yes, ma'am.
Any doubt till now?
No, ma'am.
Just a second.